Good evening, everyone. A quick indication that you can hear me, please. Does that mean no one can hear me?
Hi, everyone. Just an indication in the comments if you can hear us, please.
Okay. Thank you. Before we start tonight, we'd just like to invite you to join us in a few moments of silence in solidarity with the global fight against gross injustice and prejudice. Okay. As a quick introduction, my name is Siobhan, and Julianne, my colleague, will be online with us tonight. And you can keep an eye out for Darby in the comments. We are all from the Neural Engineering Research Venture, and we are very excited to welcome you here tonight. We have an announcement tonight, the ABC meetup, AI Brains and Computational Thinking. This is a Cape Town-based meetup open to everyone to share and explore AI's impact on the world as we further our understanding of how machines learn and think. Before I hand over to Julianne to do the introduction, the house rules. Please remember that we all come from very diverse backgrounds, and in order to create a safe environment, please use inclusive and respectful language. We would love to have you join us at the end of Betony's talk for some questions. So please post your questions in the Ask a Question feature and then upvote your favorite questions. We will be asking the most popular questions first. And as I said, we'd love to have you on stage, but if there's any problem with your mic, you can just add please ask to the question and we will ask on your behalf. If there's any problem with your browser or the streaming speed, refreshing really does help. However, if it is too slow, then the talk is available to stream at the same link. And I will now hand over to Julianne, who will introduce our speaker.
Hi, everyone. It is my great honor tonight to welcome Betony Adams as our speaker for the evening. Betony is currently doing a PhD in physics, and her research looks at the possibility of quantum effects in biological systems, a field that has been successfully applied to the phenomena such as photosynthesis and avian migration. She is currently focusing on recent evidence that quantum processes might take place in the brain. And this evening, she's going to tell us a little bit more about her research in this field. So yeah, without further ado, I would now love to welcome Betony to the stage.
Thanks, Julianne. I'm just sharing my screen. Can you see the screen?
Yes, it seems like it's working here.
Great.
Okay, I think if you adjust the aspect ratio, I don't know if you are able to now. Is it full screen at the moment?
Yeah.
Oh, there we go. There we go. Perfect. Okay.
Okay. Hello, everyone. I'm Betony Adams, and as Julianne lets you know, I'm doing my PhD in the Center for Quantum Technology at UKZN under the supervision of Prof. Francesco Petruccioni and Dr. Ilya Sinaisky. Our group is probably better known for quantum machine learning, but we also do some quantum biology, which I'll be talking about today. Thank you very much to NERV for inviting me to speak. And particularly about a subject which is sometimes seen as controversial. That is whether quantum effects may play any role in how the brain works. I've heard it said fairly often that just because quantum mechanics and consciousness are both difficult to understand doesn't mean that they are necessarily related. I'm not going to be focusing all that much on trying to explain consciousness today. Rather, I'm going to present evidence for whether quantum effects might be implicated in the physiology of the brain or the nerves that make up the brain. I won't be going into the specifics of my own research, but will rather give a broader introduction to the subject in the hope of inspiring some of you to look it up. So my talk will be broken down into giving a basic framework for the discussion by starting with a brief introduction to quantum physics and the specific quantum effects that I'll be talking about. And I'll then introduce what I mean by quantum biology and quantum neurobiology and how the two are related before I discuss some of the concrete examples to do with general anesthetics, neurotransmitters, and neural entanglement. And then I'll end off with my favorite part of the presentation, which is some interesting applications and why I think this field is really cool. So for any of you who have some idea of the history of quantum physics, this will probably all look very familiar and possibly simplistic. And all of you should recognize at least one of the faces on this timeline. But my aim is to only give you a general idea of the development of quantum ideas without getting lost in the details. And indeed, if the only thing you take away from this slide is that some of these people wanted to explain anomalous experimental results from the interaction of light and matter, and some of these people then took those observations and turned them into mathematics, then that's good enough. And it could be said that quantum mechanics developed as a way to explain experimental results that couldn't be explained using classical physics. For instance, in order to explain black body spectral energy distribution, Planck in 1900, it was known as the ultraviolet catastrophe, he introduced the idea that interactions between matter and electromagnetic radiation are quantized, with H, or Planck's constant, used as a measure of this quantization. And some of you should probably recognize this idea of quantization as being the origin of the term quantum. 5 years later, Einstein built on this idea with the quantization of light, which we know as photons, an idea he used to explain the photoelectric effect. And Niels Bohr's 1913 model of the hydrogen atom, which replaced classical electron orbits with quantized energy levels, and Compton's work on X-ray spectroscopy, all contributed to the building of the quantum era in modern physics. While Planck and Einstein began this revolution by hypothesizing that light or electromagnetic radiation, which was conventionally understood to be a wave, could sometimes act like a particle, de Broglie in 1923 had the complementary suggestion that matter might behave like a wave, which was demonstrated by the interference patterns of electrons. Quantum mechanics, then, which began as a way to explain experimental results that defied classical theory, was later formalized by physicists such as Schrödinger and Heisenberg, who gave us, along with other physicists, gave us the maths that we use to describe and predict the dynamics of microscopic quantum systems with such accuracy today. So during the course of this talk, I will probably make use of the terms quantum system, quantum state, quantum model. I thought it would be useful to explain what I actually mean by these terms. When I talk about a quantum system, I'm talking about the thing itself that I'm interested in. For those of you who did physics at school, you might remember endless questions about bricks on pulleys or cars accelerating along roads, or how the conservation of momentum relates to a collision of vehicles. So the system in that case might have been brick on the pulley or the colliding vehicles. In quantum physics, the focus is microscopic. So my system might consist of electrons or photons or atoms and interactions between these. The quantum state then describes the physical properties of a quantum system. Very simplistically, just as your car has energy or momentum that can be measured, your quantum system, electron or photon or atom, has similar properties that are described using their state vector. What is interesting, though, about quantum mechanics is that for two states describing a system, and say, for example, these are spin states, and the system could be in a spin up or spin down state, then a linear combination of these two states also describes the system. Take, for example, the state that's have on the slide. The state vector could describe an electron, which has some probability of being in the spin-up state and some probability of being in the spin-down state. This is known as the superposition principle, which might be familiar to you from the Schrodinger's cat thought experiment, where the cat is in a superposition of alive and dead. So finally, what do I mean when I talk about quantum models? Well, this is just a way of describing, using mathematics, what happens to a state over time. How does it change? If you were looking at a classical system, like a car, you might use Newton's equations to describe how the car's state changes. In quantum mechanics, you would probably use Schrodinger's equation or other related models developed over the history of quantum mechanics to describe how, say, the state of an electron changes. Now that I've hopefully clarified what I mean by system, state, and model, I want to be clearer about what are these quantum effects I'm actually talking about. So I've already told you about Niels Bohr's 1913 model of the hydrogen atom, where he postulated that the hydrogen atom or atomic structure is described by quantum mechanical principles. So in this sense, all biology could be called quantum mechanical because all biological organisms are made up of molecules and atoms obey quantum mechanical principles. But this is what people studying quantum biology call trivial quantum effects, because they don't really let you say anything new or interesting about biological systems. What we are more interested in is what you might see in papers called non-trivial quantum effects. I'm not going to go into great detail about what these effects are, but I wanted to introduce them so you recognize them when they come up later in the biological and neurobiological context. So the first of the three, I'm going to discuss three main non-trivial quantum effects. The first of these is quantum coherence. I've already described to you that quantum systems can be in superpositions of two or more different states, while coherence describes the relationships of states in a superposition. Quantum tunneling, which I will also discuss in the context of biology, is the non-zero probability for a quantum particle to pass through a classically forbidden energy barrier. And finally, I'll be talking about entanglement, which is the non-classical correlation between two quantum particles. So if any of you have done any reading on quantum biology or quantum computing, you will have no doubt heard about this thing called decoherence, which refers to the loss of quantum effects in a system. For this reason, quantum systems have conventionally been described as isolated and at low temperatures, because interaction with an environment, particularly an environment that gets messier as the temperature increases decoherence. But biological systems probably by definition have to interact with their environments, and they exist at physiological temperatures. So it's impossible to use models that are developed for conventional quantum systems to describe biological examples. But physicists have already developed a way of modeling how a quantum system might interact with an environment, and this is known as open quantum systems. My supervisor, Professor Petruccioni, wrote one of the seminal books on the subject. The open systems approach is used by many research to describe quantum effects in the biological context. To simplify, the system under study is broken down into the system of interest, which might be electrons moving through a leaf, the environment these electrons interact with, and then might be modeled as harmonic oscillators, and then the interaction between the two. So what exactly then is quantum biology? The question of whether physics might contribute to understanding biology is not a new one. And the question of whether quantum physics might contribute to understanding biology has a history almost as long as quantum physics itself. Schrödinger's 1944 lecture series, What is Life? The Physical Aspect of the Living Cell, is often used as a milestone in quantum biology. In it, he describes an aperiodic crystal responsible for hereditary traits, and this later inspired Watson and Crick in their studies of DNA. Before this, however, Bohr had already given a lecture called Light and Life, and other physicists such as Pascal Jordan had already coined the phrase quantum biology. Of the non-trivial quantum effects I will discuss in this presentation, quantum tunneling was the first to make an appearance in the 60s already in the context of enzymes. A decade later, coherent spin dynamics in radical pairs was proposed as a way to describe magnetoreception. And last, but experimentally most successful of the topics that today are recognized as quantum biology is coherent energy transfer in photosynthesis. Pretty much the main objection raised against the possibility that quantum effects might play a role in biological systems is the phenomenon of decoherence. As I've mentioned already, quantum effects are delicate and easily destroyed by interaction with their environment, whereas biological systems unavoidably interact with their environments. But the field of quantum biology, as we understand it today, has made some progress towards establishing that quantum effects might play a role in biological systems. And it has arguably had most of its success in the context of three different biological phenomena: photosynthesis, olfaction, and magnetoreception. In the case of photosynthesis, energy and electron transfer in plants has been suggested to employ quantum coherence. This, despite there's still some contention over this, but it seems to have been supported by experimental findings. Quantum tunneling has a long history in quantum biology, having first been observed in enzymes. Its most famous application is arguably to olfaction, or the sense of smell. And finally, the spin dynamics of radical pairs in birds' eyes have been used to explain some of the behavioral aspects of the avian campus. And it's on this foundation that quantum neurobiology is being built. So finally, we get to the neurobiology. I'm sure there are many of you here tonight who have a far more nuanced understanding of neural structure and function than I do. This diagram of a nerve cell is only a useful way to locate the quantum effects that I'll be talking about in a neural context. So the parts of the nerve that I will be referring to are as follows: the microtubules, which make up the cytoskeleton of cells and are important in cell structure, transport, and cell division. Mitochondria, which are the site of electron transport chains, which are similar to electron transport chains in photosynthesis. And mitochondria are also the main site of biophoton production. Between the nerve cells, the synapses, I'll be talking about neurotransmission, and synapses are also the sites at which hypothesized Posner endocytosis takes place. And then finally, the myelinated axon has been suggested to possibly act as a waveguide for biophotons. So I've given you some idea of the history of quantum biology, but what about the history of quantum neurobiology? Well, I think quantum theory has a long history of inspiring various discussions about the nature of consciousness. But the first detailed theory that outlined how quantum physics might be applied in the context of neurobiology or consciousness emerged in the 90s. Roger Penrose, who is a physicist famous for his work in relativity and cosmology, wrote a book called The Emperor's New Mind, where he suggested that classical rules aren't enough to explain consciousness. What he lacked, however, was a biological medium to locate these quantum effects. Stuart Hameroff is an anesthesiologist, and he'd been interested in microtubules. If you remember, microtubules are part of the cell cytoskeleton and are important in cell division. He'd been interested in microtubules for a while, and he suggested that they might possibly support quantum effects. And together they came up with this theory that's called orchestrated objective reduction, where quantum computations in neurons change axon firing and thus consciousness. I'm not going to go into the full, because the theory has gone through a number of iterations and it's been criticized by a number of scientists. But if you're interested in reading about it, there's a review, a 2014 review called Consciousness in the Universe, which is very comprehensive. My focus in this talk is less on consciousness than on the physiological mechanisms that allow neurons to function. What I would like to focus on from the theory of orchestrated objective reduction is the biological site that they chose to situate their theory in, which is microtubules. So microtubules are formed through the polymerization of tubulin dimers. Tubulin dimers consist of an alpha and a beta tubulin protein, and they form filaments, and then 13 of these filaments form a microtubule. What was interesting to Hameroff and Penrose about tubulin dimers is that they are potentially a way to realize biologically a qubit. So what do I mean by a qubit? For those of you familiar with computer science, you'll know that a binary bit can have a value of 0 or 1. A qubit, on the other hand, is a quantum bit. And as we've already heard in this talk, quantum states can exist in superpositions. So in this case, maybe 0 and 1. When orchestrated objective reduction was first developed, Hameroff and Penrose suggested that tubulin dimers could exist in a superposition of conformational shapes, but the theory has since been updated to include electric and magnetic dipoles. But as I say, if you're interested, there's the 2014 review. I'm not going to say anything more about orchestrated objective reduction, but I will get back to microtubules in a bit. So I like this quote because it sums up, at least for me, one of the methods by which it's possible to address the unsolved problem of consciousness, or on a lesser level, the physiology of the brain, and that is to study those chemicals or interventions that have an effect on consciousness or change brain function. And this approach will inform, to a large extent, my presentation as I discuss research relating to general anesthetics, to neurotransmitters, and to antidepressants such as lithium. So this image is also borrowed from literature, and what it highlights is the extreme diversity of the various chemicals that act as general anesthetics. So as you can see, they range from very simple noble gas xenon to the unwieldy looking olfaxolone. And what is remarkable is there doesn't seem to be a uniting factor or feature, which is what led Lukaturin to hypothesize that maybe there was some other way of linking these different molecules. It was a question that Hameroff had already addressed to some extent in wondering that whether anesthetics were not chemically active, but electronically active. So what do I mean by electronic? Well, very simply, in this case, I mean having something to do with electrons. A large part of the success of quantum biology has been in describing how energy or electrons move through a biological medium, such as a leaf, during photosynthesis. In particular, it has been suggested that this movement uses quantum coherence. To simplify things, at least as how I understand it, what this means is that when energy is transferred between pigments known as chromophores, which react to light, the energy is shared or delocalized over sets of chromophores. So in the diagram, the chromophores are the molecules in that black circle, and each colored semicircle represents chromophores that are sharing the energy, whereas between the colored semicircles, the energy is moving classically. So if you take anything away from this slide to apply in the context of neurobiology, let it be that the arrangement of these pigments called chromophores in a protein allows for this coherent energy or electron transfer. What I'm trying to illustrate with this slide is how quantum neurobiology has borrowed from work done in quantum biology. Once again, we're interested in the movement of energy or charge through a protein, but in this case, our biological context is microtubules in nerve cells rather than antenna complexes in leaf plants. Tubulin proteins, which make up microtubules, have similar arrangements of chromophores to photosynthetic networks. This has led researchers in quantum neurobiology to suggest that not only do microtubules allow for coherent energy charge and charge transfer in nerve cells, but that anesthetics bind to these proteins and disrupt this coherent energy transfer, and that this is the mechanism of action of anesthetics. So is there any experimental evidence for anesthetics being electronic rather than chemical agents? Well, Turin, whose quote began this section, was also interested in whether the movement of electrons in proteins was affected by anesthetics. To test his ideas, he focused on Xenon, which is the simplest of the anesthetics, and as he argued, doesn't have shape or chemistry, but has physics. For inspiration, he used an experiment done by IBM, where a scanning tunneling microscope was used to write IBM using single atoms, where the xenon atoms extend the conducting surface of the metal. In the same way, Turin argued, xenon atoms binding to proteins could extend the highest occupied molecular orbit and allow for electron transfer in proteins and facilitate electron currents. In order to test this, he looked for an increase in free electrons in fruit flies under the influence of anesthetics. The free electron signal was measured as an increase in spin using an electron spin resonance experiment. Spin is the attribute that particles like electrons have that makes them behave like little magnets in a magnetic field. So applying a magnetic field causes a split in possible spin energies. And then if you introduce a radio frequency electromagnetic source that matches this split in energy, then the radio frequency radiation will be absorbed, and the amount of absorption will tell you something about the number of spins. And what they found was that anesthetics do cause an increase in spin, which possibly corroborates this hypothesis that general anesthetics cause electron currents in proteins. However, there is still some contention as to where this free electron signal was coming from. It could be coming from melanin. Also, the fruit flies were basically frozen and starved of oxygen at the same time. So it's, yeah, the experiment probably needs to be redone with more physiological environments. So I've mentioned quantum tunneling a number of times now, and in the context of enzymes as well as olfaction, or the sense of smell. To give you a better idea of what I mean by this, I thought I'd discuss the concept in the context of quantum biology before moving on to the neurobiological context. So the lock and key or docking mechanism has been used to describe the working of a number of biological systems, enzymes and olfaction included. And I realize that to some extent this is a simplification, but I'm not a biologist, so any biologists feel free to correct me. The basic idea is that the shape of a molecule, such as an odorant or smell molecule, matches the shape of a specific receptor, and the correct fit activates the receptor. But it has also been suggested that odor receptors, which are known as G protein coupled receptors, might be activated by the vibrational modes of specific odor molecules using vibration-assisted quantum tunneling. Very simplistically, in order for a receptor to be activated, an electron needs to move from one level to the next. If the vibrational energy of the odorant molecule matches the energy of the transition, then the electron moves levels and the receptor is activated. So if you remember nothing else from this slide, then hold on to the fact that lock and key mechanisms could potentially actually work through vibration-assisted quantum tunneling, which we will now look at in the neurobiological context. As I've already mentioned, olfactory receptors are GPCRs. GPC receptors are everywhere in the body. I think something like 1/3 of all pharmaceuticals are targeted at these receptors. And the receptors that neurotransmitters bind to on nerves are also GPCRs. Many of you probably know more about neurotransmitters than I do, but as a quick summary, neurotransmitters are chemicals that allow nerves to communicate at synapses. The binding of neurotransmitters to their appropriate receptors controls the movement of charged ions across nerve cell membranes and causes changes in potentials and the firing or not firing of neurons. Following from work done on olfaction, there's now been some suggestion that neurotransmitters such as serotonin and histamine and adenosine bind to and activate receptors through vibration-assisted quantum tunneling and not lock and key mechanisms. This work is still very theoretical. For example, agonists of the serotonin system that activate the same receptors as serotonin, such as LSD, share similar vibrational spectra as calculated by density functional theory. While the research is very much still theoretical, I think there've only been 1 or 2 experiments to try and prove it. There have been some experiments done, and the method of testing this theory follows from the example of olfaction, or the sense of smell, which uses selective deuteration. Selective deuteration means replacing hydrogens in your molecule with its heavier isotope deuterium, which has an extra neutron. And there's some evidence that species as various as fruit flies, cockroaches, and whitefish can smell the difference between hydrogen and deuterium. And this would support the case for vibration-assisted quantum tunneling. Selective deuteration changes the mass of the molecule, thus changing its vibrational spectra, but it doesn't change the shape. So how have neurotransmitters fared in similar experiments? Well, serotonin, which was a good contender based on the theory, which showed that agonists of the serotonin system shared vibrational spectra, unfortunately didn't perform very well in experiments, though I think there's only been 1 experiment done. But histamine, in the selective deuteration, did make a difference to the action of histamine, though there's some argument over whether this is due to vibrational assisted tunneling or some other mechanism. More experiments need to be done, as selective deuteration is a delicate operation, and vibrational spectra can also be thrown out by experimental mistakes like introducing microscopic amounts of contaminants. So I've discussed 2 out of 3 of the main phenomena associated with quantum biology, and how progress made in understanding these biological processes has been applied to understanding related neurobiological phenomena. In this 3rd case, we are interested less in the movement of electrons than in their spin dynamics and how spin might translate into chemical or biological signaling. To recap, spin is a property that particles such as electrons or nuclei have that makes them behave like magnets in a magnetic field. So it's been known for quite a long time that spin dynamics can have an effect on chemical reactions, and this was adapted to try and explain birds' incredible navigational feats. The model that was developed is called the radical pair mechanism, and it can be basically summed up in 3 steps. First, a photon, so a light particle, transfers its energy to an electron in an electron pair, causing that electron to be donated from a donor to an acceptor. This means you now have a spatially separated but spin correlated electron pair. And what I mean by spin correlated is that the spins of each of the electrons are arranged in a specific way with respect to each other. In this case, theory has it that the spins start off in what is known as a singlet state. Then, under the influence of the Earth's magnetic field and the surrounding nuclei, this singlet state interconverts between singlet and triplet state. Now, if recombination happens when this spin state is in a singlet or a triplet, you get different chemical products, and in this way, you can have a spin-dependent physiological outcome. So how does this radical pair mechanism offer any insights into quantum neurobiology? Well, one of the hypotheses that has been put forward in the field also depends on a spin-mediated physiological outcome, in this case using that most mysterious of quantum phenomena, which is entanglement. In this case, unlike the radical pair mechanism, we are interested in nuclear spin, not electron spin, but the idea is similar. So physicist Matthew Fisher was interested in whether quantum physics could play a role in correlated neural firing patterns. And he hypothesized a way in which entangled nuclear spins of phosphorus nuclei could result in entangled neural activation using molecules known as Posner clusters. I'll be discussing this in further detail in the next few slides, but what I wanted to highlight here is that once again, we are looking at how spin correlation can result in a physiological outcome or biological outcome. So how did Fisher get interested in this subject? Well, as the story goes, he had a period of severe depression, which got him interested in antidepressants and mood stabilizers, such as lithium, which is used to treat bipolar disorder. Similarly to the argument that Turin made about Xenon, lithium is one of the structurally simplest antidepressants. And Fisher hypothesized that if neural processes use quantum effects, then drugs that target neural processes might have different neurophysiological effects for different isotopes. So he did some research, and if you recall, isotopes are variations of elements that have added numbers of nuclei. He did some research and found evidence that experiments had been done using lithium isotopes, lithium-6 and lithium-7, which showed that lithium-6 promotes maternal behavior in rats far more strongly than lithium-7 for the control group. More recently, a similar effect has been shown for xenon isotopes. If you recall, xenon is the simplest of the general anesthetics. It has 9 stable isotopes. 7 of these have 0 nuclear spin, but 2 of them have non-zero spin. So xenon 129 has spin half, and xenon 131 has spin 3/2. And researchers found that they used writing reflex as a measure of consciousness. They found that xenon with non-zero nuclear spin was a less potent anesthetic than those with 0 nuclear spin. I personally think this is an incredible result. Something as tiny as adding a neutron can have such a profound effect as to alter states of consciousness. If you've been paying attention and you recall our previous discussion on isotopes in the context of neurotransmitters, you might be thinking that these isotope-dependent effects have something to do with mass and thus vibrational spectra. But the researchers checked, and the results weren't dependent on atomic mass, which means that they most likely have something to do with spin. So this interest in spin led Matthew Fisher to hypothesize that spin-mediated reactions might play a role in neurons. But first he had to decide on which element would be best suited to act as a neural qubit. I've talked about decoherence before and how it causes loss of quantum effects. In order to have any effect on neural firing, or even more ambitiously, memory storage or retrieval, Fisher needed a qubit that where decoherence happened really slowly. So to give you some idea of the timescales of quantum effects in biological contexts, this slide will show you in photosynthesis, coherent transfer and photosynthesis happening on the order of picoseconds. For the avian campus, which is the electron radical pair mechanism, the coherence lifetimes are microseconds. Nerves are approximately milliseconds to seconds. But plasma molecules, which Fisher postulated, could have coherence lifetimes of seconds to hours. So how did Fisher propose to achieve quantum effects lasting for hours? To do this, he settled on phosphorus nuclear spin as the physical site of his proposed quantum effects. Phosphorus has a nuclear spin of a half, and for spin half nuclei, decoherence happens much more slowly because it only happens through interaction with other nuclear spins and not with electrons through the quadrupole moment. Quadrupole moment of spin half nuclei is 0. There are not so many common biochemical elements with spin half, hydrogen and phosphorus, so that's how he settled on phosphorus. And he calculated that phosphorus could maintain its coherence if decoherence was happening only through intermolecular interactions of these plasma molecules with surrounding extracellular fluid, then they could maintain coherence for 21 days. There has been some controversy over that very long time, with other researchers suggesting that actually this coherence would only last as long as 37 minutes, which is still a very long time for coherence to last. So what exactly is an entangled plasma molecule, and how do you create one, and how does this spin entanglement translate to neural firing? So to start off with, you need ATP, which is the molecule that cells derive their energy from. It's made in mitochondria and it's present in every cell in the body. From ATP, you get pyrophosphate, which is 2 phosphate ions. Phosphate ions are phosphorus surrounded by oxygens. The next step is the creation of phosphates from these pyrophosphates, and this is the important step quantum mechanically because this is where the entanglement happens. Fisher's theory is that if this creation of phosphates from pyrophosphate happens through the use of this enzyme pyrophosphatase, then molecular dynamics mean that the phosphorus nuclei end up in a singlet state. If you remember, a singlet state is a specific spin correlated state, so spins are entangled. Now, if these phosphate molecules interact with surrounding hydrogens, then decoherence happens very quickly. So you need a way to protect the quantum effects against decoherence. And Fisher proposed that if these phosphates bind to calciums, then the calciums have a 0 nuclear spin, the oxygens have 0 nuclear spin, so this acts as a sort of cage of 0 spin around these phosphates and protects against decoherence. So how do these entangled phosphorus nuclear spins have any effect on neural action? Well, Fisher imagines that entangled phosphates that end up in different plasma molecules might get taken up into different neurons during synaptic endocytosis. Then under a change of pH, the plasma molecules can bind, and when they bind, they melt and they release calcium, and calcium is implicated in the release of neurotransmitters. And Fisher's hypothesis is that entanglement can change the binding rates of plasma molecules, which in turn will affect the release of calcium and the downstream action of neurotransmitters. So this is all still extremely theoretical, although I think he got a very big grant to try and test it experimentally. And so while it still has to be tested experimentally, it also possibly offers a way of imagining how lithium isotopes might have an effect on neural action. It's been shown that you can replace the central calcium ion in a positive molecule with a lithium ion. And then the different, if you remember lithium, so calcium has spin, zero nuclear spin. Lithium has, well, different isotopes, so it has either spin half or spin 3/2. Well, I mean, spin 1 or spin 3/2. And these different spins affect the dynamics and possibly have an effect on the entanglement and thus on the calcium production and the neural activation. What is also potentially interesting about this proposed Posner molecule mechanism is its similarities to ideas put forward in quantum computing. One of the challenges of quantum computing is the physical realization of qubits. Various qubit alternatives have been explored, from superconducting circuits to trapped ions. And one of the first suggestions for an implementable, scalable quantum computer was made by Bruce Kane in 1998. And he proposed that information could be encoded onto the spin half nuclei of phosphorus in a substrate of spin 0 silicon, which seems quite similar to Matthew Fisher's phosphorus nuclei in their spin 0 cage of oxygen and calcium. Spin-based quantum computing is still one of the main avenues being pursued in the quest for scalable quantum computers, and particularly, I think, in Australia. So I've been through some of the details now about how quantum effects might play a role in effective neural processing, but to end off the presentation, I wanted to talk about why I find the field so interesting and potentially really useful. So I read an article the other day about the effects that serotonin is having on a species of shrimp. Serotonin, as I'm sure a lot of you know, is a neurotransmitter. In fact, apart from dopamine, it's probably the most famous of the neurotransmitters, being partially responsible for what we call happiness. Many antidepressants are SSRIs, which stands for selective serotonin reuptake inhibitors. Millions of people take these drugs. I think I looked it up. 25 million people worldwide are taking SSRIs, which means that water supplies are beginning to have measurable concentrations of SSRIs in them. And recently it was discovered that exposure to SSRIs was causing the death of a certain species of shrimp. This was because the shrimp, which ordinarily hid in darker waters, were swimming towards the light and consequently getting eaten in larger numbers. So why I find this so interesting, but unfortunate for the shrimp, is that the molecule that makes up serotonin is tryptophan. And tryptophan is one of those light harvesting pigments called chromophores that I was talking about in the context of photosynthesis and in general anesthetics. Tubulin, for example, the protein that makes up microtubules, has 8 tryptophan chromophores embedded in it. I think it's quite amazing that our consciousness, if it's mediated by neurotransmitters such as serotonin, might be in some sense photosynthetic. In 1932, Niels Bohr gave a lecture called Light and Life for the opening of the International Congress of Light Therapy in Copenhagen. So light and electromagnetic radiation was central to the development of quantum mechanics. Out of Einstein's many groundbreaking theories, it was photons and the photoelectric effect that won him the Nobel Prize. Light is also central to life, with photosynthesis being responsible for a large percentage of Earth's biomass. So it's no surprise that progress made in understanding light might have been interesting in the context of biology. But while it's easy enough to find Bohr's lecture, where he discusses what new discoveries in quantum theory might mean for the study of living organisms, it's more difficult to trace what happened to the therapy part of the Congress. The meeting was devoted to the role of light in biology, biophysics, and therapy. The role of light in biophysics has subsequently incorporated quantum theory in its study of photosynthesis. But the idea of light therapy seems to have fallen away after the initial Congress. We know for sure that plants interact with light, but it is less certain if or how humans might interact with light. So this, to some extent, has now re-emerged as a growing field of research. So quantum mechanics deals in great detail with light-matter interactions. But what can it tell us about light-living matter interactions? The diagram shown here illustrates the electron transport chains of photosynthesis and respiration. Electron transport chains are integral to living organisms as they create the electrochemical gradient that drives ATP production. And as I've mentioned before, ATP is the molecule that cells derive their energy from. As you can see in the bottom section of the diagram relating to photosynthesis, light is harvested by certain complexes in the electron transport chain, and this energy is transferred to the chain, through the chain, to result in ATP synthesis. The top diagram is a similar electron transport chain that is found in mitochondria, present in all of our cells. And these similarities are potentially interesting in the context of transcranial photobiomodulation, which is the application of red or near-infrared light to the cranial area. The proposed target of this light has been suggested to be one of the complexes that make up the electron transport chain in mitochondria. Is it possible that in some sense we also have biological apparatus for photosynthetic mechanisms, and can quantum mechanics, which has contributed somewhat to understanding photosynthesis, help explain light-matter interactions in a similar context in mitochondria? Light therapies such as photobiomodulation have shown success in treating a variety of brain disorders, ranging from depression and schizophrenia to brain damage. It's also been shown to improve memory, learning, and executive function. South Africa is not so behind the trend either. I recently saw that Carte Blanche had done a show on a Durban neurologist who has successfully treated patients with Parkinson's and brain damage using photobiomodulation. Quantum dots, which are nanoscale semiconductors with quantum properties that can emit light, have also been shown to be a promising therapy for undoing the protein tamping that causes Alzheimer's and Parkinson's disease. What I find even more interesting is that not only might we harvest photons, but our cells actually produce light, biophotons, which are ultra-weak, near-ultraviolet to near-infrared photons. So the site of biophoton production seems to be the mitochondria, and though it is still a new area of research, there's some suggestion that biophotons may be an alternative form of neural signaling, with researchers hypothesizing that the myelinated axon could act as a photon waveguide. So do biophotons offer us any insight into the brain? Well, Sylvia Plath wrote during one of her periods of mental illness, "This is the light of the mind, cold and planetary. The trees of the mind are black, the light is blue." Recent research, however, suggests that the light of the mind is not blue, but actually red. An experiment found that when slices of brain from species ranging from bullfrog to mouse to chicken to pig to monkey to human, not necessarily Marie Curie, were stimulated with glutamate, the frequency of the biophotons they emitted ranged according to the intelligence of the species. While the measure of intelligence is still debatable, and correlation does not mean causation, it appears as though higher intelligence corresponds to redshifted, lower frequency biophotons. Biophotons are also related to reactive oxygen species, with mitochondrial reactive oxygen species hypothesized to be the source of metabolic biophotons. Reactive oxygen species are very important in cellular signaling, but they can also cause great damage to, they're very reactive and can cause great damage to cells. They're implicated in a variety of damage, from DNA damage to disease, mental illness, and aging. And when people talk about antioxidants, it is reactive oxygen species that these antioxidants are aimed at. Which brings us to another way in which quantum mechanics might contribute to our understanding of the brain. So I've spoken about the quantum property of spin a number of times already. but it also appears that spin dynamics might play a role in reactive oxygen species production and control. So it's generally accepted that a number of animals have a magnetic sense, but it's unclear, it has been unclear whether humans have any way of sensing weak magnetic fields like the Earth's magnetic field. But a recent experiment showed that changing geomagnetic fields disrupted alpha waves, which are 8 to 13 hertz oscillations in the human brain. It has also been shown separately that biophoton increase is linked to alpha rhythm fluctuation. As biophotons are linked to reactive oxygen species, is it possible that reactive oxygen species play some role in this magnetic sensing? If you recall the discussion of the radical pair mechanism of the bird's compass, I talked about the switching of spin states between singlet and triplet states. And it's been shown that with respect to reactive oxygen species, this singlet triplet mixing can influence the production of reactive oxygen species. And it has also been shown that magnetic fields at Earth strengths can alter the spin mixing and thus change reactive oxygen species yield. What is potentially interesting about this is that there's some evidence that geomagnetic storms, which cause fluctuations in the Earth's magnetic field, have been correlated with psychological effects such as increased rates of suicide. Whereas this needs far more research before any concrete conclusions can be drawn, reactive oxygen species might be an interesting way of explaining these results. Quantum mechanics is often adopted as a quasi-mystical theory open to all sorts of misinterpretations. What I thought was interesting, however, is that there is some evidence, though probably not nearly enough to base any conclusions on, that meditation decreases biophoton emission. And could this point to a decrease in reactive oxygen species and thus explain the beneficial effects of meditation? That's as much as I'm going to say about how quantum effects might play a role in the physiology of the brain or how nerves function, but I thought I'd end off with a very brief discussion of how we might extend the discussion to address human behavior. Niels Bohr, who you've heard a lot from today, already felt strongly that the formulation of quantum theory might have something to offer on a more philosophical level. And indeed, quantum cognition has made some interesting inroads into accurately modeling human behavior. Very briefly then, quantum probability models have been shown to more accurately model behavior that is seen as irrational. The example that I found most interesting was what is known in psychology, or apparently known in psychology, and feel free to correct me as it's not my speciality, as context effects. And that is that the order in which you ask certain questions matters. For instance, if you ask people first whether Bill Clinton was a good president and then whether Al Gore was a good president, they give different answers if you switch the order of the questions. Why this is interesting in the context of quantum theory is because of commutativity, which represents the order in which you measure physical observables and is a fundamental principle in quantum theory. I'm not going to say more about this, as it's not my field of research, but I think it's extremely interesting for those of you who want to go read more about it. And for those machine learning experts listening, there was also a recent experiment that showed that quantum reinforcement learning models were better at modeling value-based decision-making on both a psychological and a neural level. With that, I'm going to conclude with a very open conclusion, because most of what I've discussed is purely theoretical and far more experimental evidence is needed before any conclusions are drawn. But I think it's a really exciting field that could potentially lead to all sorts of new therapies for brain disorders. It might tell us something about human behavior and, who knows, ultimately perhaps consciousness. Image reference list. And yes, that's the end of my talk.
Thank you so much, Betony. This was a fascinating presentation. And we have some very exciting questions. So we have about half an hour left. So I think we will start with the questions now. And then we will see through how many we are able to get within the time we have remaining. So first on screen, we have Lee, who will be asking his question.
Hi, I'm Lee. I'm a postdoc in UCSF. Thank you for your fascinating talk. I'm always hooked by quantum physics. And I think there are many complex questions and fascinating questions like the mechanisms of consciousness and the anesthetic effects is actually linked to the human conscious. So my question is about, as most anesthetics affects to GABA receptors, so is there any control experiment saying that most anesthetics, indeed, all of them can increase spin, and any control experiments indicate that the chemicals that cannot increase spin, they may not have anesthetic effects?
So you mean, have they tried experiments with other, so related, not anesthetics, but related, and see if they cause them? As far as I know, they've only done the experiments for general anesthetics, but I think that would be something interesting to do, to see if you can get maybe a lesser. I think they did link potency of anesthetics to the measure of spin increase. So that is sort of some way towards that. But yes, it would be interesting to use completely unrelated, like maybe psychedelics, and see what happens to spin.
Very fascinating. Thank you.
Okay, so we have our next question from Taliesin, and I just invited him on screen, so I think let's just give him a moment to join us. Okay. Okay, there we go. He should be coming on shortly. Thank you for your question, Lee.
Yeah, thank you.
There we go.
Hold on. I've lost audio. Hold on a sec. I can't hear anything.
Okay, we can hear you clearly. Let me just comment that quickly. Okay, can you still not hear us? I'm guessing. Okay, let me just quickly see if we can. Oh, you know what I'm going to do? I'm going to ask the question so long. Then Betony, you can perhaps start answering. So Taliesin wants to know, has the Xenon result been replicated?
No, not as far as I know. It's quite a recent experiment, and unless it's been replicated in the last few months, then no, they haven't. They haven't looked at. You mean oh, which one are you talking about? The Xenon electron spin, or the Xenon with the different isotopes?
The Xenon with the different isotopes.
That, as far as I know, hasn't been replicated. No.
I did briefly just look at his recent publications, and it did look like there was an attempt to replicate the lithium experiment.
Which lithium? Oh, the rats. No, that didn't give any. Okay.
And it sort of worked. Like they had to inject ketamine. Like it didn't work, and then they injected ketamine, and then it did work. And it was like, why did you inject ketamine? Was that the original plan?
Okay, great. I'll have a look. Thanks. Because my research actually is on lithium, so lithium spin. Cool. Thanks.
Okay, thank you so much for your question. Okay, next up we have get here. So we have Patricia. So I'm just inviting them on screen. Okay. It's great that we have this sort of online interaction tonight. That's awesome. Okay. Go. Oh, my apologies. I see Patricia says, please ask. Okay, so I will then quickly ask this question on their behalf, and then we can get to answering it. So Patricia is asking, why argue that anesthetics must have a common mechanism of action? AF, AI, Chi, there are many different, anesthetized states. That is, many different ways of disrupting consciousness. Different molecules can be affecting the brain in so many different ways, with an apparently common end result, right?
That's a good point. I mean, maybe I can ask the question back, is how do different, I thought, as far as I understood, general anesthetics had a fairly similar action on bodies, is that they kept certain physical, you keep breathing, but your consciousness disappears. If you can you explain more what you mean by what are different levels of being under general anesthetic?
Okay, we can just see in the comments.
I think also for Lucatorin, it was just a way of starting, a way of trying to find some way of having a mechanism, because it would be a good guess to think that if these molecules all cause unconsciousness, then they possibly are working through the same mechanism.
Okay, that's good. I think we'll leave that answer at that. Next up, we have Andre's question, and I am inviting Siobhan on. She should be on shortly to ask on his behalf. Oh, there's a comment from Patricia.
Yes, I agree. They definitely are different, and I think they measured that as a level of potency of anesthetics. They do have different effects, and they did have different. So they did have different amounts of spin. So the had spin. So some anesthetics created a greater or lesser increase in spin, but they all seem to have this common increase in spin. So that would suggest that was what was causing the lack of consciousness. But as Lee, I think, suggested, it would be interesting to maybe try other molecules that have an effect on consciousness that isn't general anesthetic, and see what happens to, does it also cause increases in spin?
Okay, perfect. Okay, another comment there.
Okay, I don't know enough about different anesthetics, to be honest, to be able to answer that question. So sorry.
Okay. So it seems like we have a bit of difficulty inviting Siobhan on. So I'm just quickly going to read the next question that we have from Andre. So Andre is asking, or first he's saying, very nice talk. And he's saying, I'm a physicist working on ADS, CFT duality and holography, and want to ask if the sort of error correction that gravity in the bulk is responsible for in ADS/CFT might be used as a paradigm for imaging robust quantum computations done in the brain. After all, the brain is structurally a surface bulk, and computation is done on the surface, exactly like in ADS/CFT. Do you think holographical thinking may be useful in the context of neural processing?
Goodness, I think I'm unqualified to answer that question because I'm going to have to ask you what holographical thinking is. Sorry.
Okay, let's see. Andre was in the comments. I'm sure he's still here. Give him a moment.
Yeah, I'm not sure I'll be able to.
Okay, let's see. Yeah, it seems like it's quiet in the comments. So we're going to leave this question here. Then Charlison is asking another question. So, oh, there's a comment from Andre now. He's saying, sure, it's the idea that one has a duality between a theory living on the boundary and a corresponding theory living in the bulk.
I think most of the theories I've put forward here have been things that are happening inside single neurons. So electron transfer in microtubules in a neuron, neurotransmission by vibrational tunneling. So I haven't ever really considered, I'm probably considering, I haven't considered what the bulk qualities would be. So I'm not sure I could actually answer that question, if that makes any sense.
Okay, moving on. So here we have Taliesin again to ask his next question.
Yes, so didn't really think I would ever be saying this kind of thing because I'm not that kind of person, but I was kind of somewhat surprised to find out that there is actually quite a body of evidence, experimental evidence built up over several decades that what we thought was impossible, which is that non-ionizing radiation doesn't have any significant thermal effect on biological tissue, can actually influence biological processes and make things go wrong, right? And have deleterious health effects. And that's been replicated for the frequencies that are in use even today with Wi-Fi, cell phones, etc. And like, I mean, I still have my iPhone here. I still enjoy Wi-Fi. I'm obviously not too freaked out about it, but it's certainly not something that's in the fringes. I mean, it's something that like the researchers who study this are pretty confident about, and it's replicable. And
yeah,
there seems to be a sort of consensus policy response that the public isn't prepared to really change their ways, even if it is harmful to the public health. And I just thought, what do you make of that? And do you think that some of the research that you're presenting sort of bolsters the idea that there could be
I think
could be bad things going on all around us, right?
I think it's a really interesting question. And I did my master's on bird migration, and there was a study, I think in 2014, where they found that very broadband, very low, way below the World Health Organization radio frequency radiation disrupted bird migration. The birds couldn't migrate, or they couldn't orientate themselves. And this was it was a very well replicated result. But what was interesting to me is that it didn't seem to be a long-term effect. As soon as you took the birds away from that radiation, they were able to use their compass again. So I don't know if maybe if they'd been in it for a long time, it might have changed things. But what I do find interesting is that the molecule that is in the eye of the bird, where this radical pair takes place, is cryptochrome, and cryptochrome is also responsible for circadian rhythms. And there's been some argument. I think there's a paper called magnetocarcinogenesis, where they argue that throwing out by this broadband radio frequency radiation throws out circadian rhythms, and throwing out circadian rhythms has been linked to things like depression and cancer. But then the same guy who collaborated on that, he also wrote a paper saying that actually the effect of, say, like power lines or the radiation coming off it would be similar. It would have a similar effect to like moving a kilometer up in the magnetic field. So, which doesn't seem to would change the radical pair and disrupt the circadian rhythm in the same way as maybe flying to another country. But another thing that's interesting is that they might also use these things therapeutically. They've found that radio frequency, and my research is looking at these plasma molecules, and they have these transition frequencies between these spin states that are of like hertz up to like 5,000 hertz. And they found, and plasma molecules are precursor to bone, or hydroxyapatite, which is the precursor to bone. And they found that it's called nano-kicking. Nano-kicking stem cells at these frequencies can turn them into bone cells. So you could also be using it as kind of therapeutic things. So I think understanding it would be, very instructive.
Pretty wild.
They're such tiny effects. So I think, like as I was saying about geomagnetic storms, I mean, it's such a small effect that I'd say you already have to be sort of on the brink, and then maybe this just tips you over. But yeah, I think it's a very interesting thing to think about. So if we have all these,
I might start sleeping with a tinfoil pillow.
So don't take me as the expert, please.
Okay, then we have another question from Taliesin. So I'm just going to ask you to ask that one also while we have you.
I can't remember what the question was.
Okay, I will read it to you. It says, I spend most of my time in a dark room looking at a tiny white screen. Is this bad for me?
Somewhat
of a joke, but I suppose it is actually a serious concern.
Well, what I'm looking at so if we are full of these chromophores, then I mean, so the circadian rhythm one is so birds migrate using blue light. So cryptochromes are blue light activated. So if you're looking at blue light, you could be messing with, as I said, prototype activation with red light, and that's been shown to be therapeutic. So it depends on what colors you're staring at, maybe. But again, I'm not the expert. But I think light matter interactions are really fascinating. I know I read somewhere that they are looking at using a specific frequency of green light for migraine treatments. So,
wow, cool.
Perfect.
Thanks.
Okay, awesome.
Siobhan.
Thank you for your question. Okay, so I'm just going to ask Siobhan to read our final question out while I'm inviting our attendee on screen.
Sorry, is Adriana coming on screen?
She says, okay, her video may not be good enough. Okay, let's just stick to you asking it then. I think that might be our best bet.
Okay, great. Yeah, we definitely have time for the last two questions. So a lot of discussion has gone on around the health implications of 5G. Does your knowledge on light therapy or the impacts on biology shed any light on this concern? On millimeter wavelength light in our living spaces. I hope I read that correctly.
I think that was pretty similar to what we were just talking about in terms of the radio frequency, broadband radio frequency. So like I think in birds, it was from kilohertz to megahertz, and very low intensities as well, disrupted birds' capsules. So that's a small signal that actually threw out a biological mechanism. So yes, I think they definitely had an effect, just one effect, because as I was saying, once you took those away, it was fine, it wasn't a lasting effect. But I definitely think that it does demand more research. So.
Okay, then our final question is directed at you. It's from Davi. And he's asking, so we did introduce you, but maybe you could tell us a bit more about your own research in your own words. And then if you could explicitly state whether your research is experimental or theoretical.
Okay. Well, if you remember the plasma molecules, which are these molecules with the entangled phosphates or nuclear spins that somehow cause correlated nuclear firing. And what I'm interested in doing is replacing the central calcium with lithium, and then looking to see whether maybe I should first say that it's very much theoretical, because I'm not. I don't think anyone's ever actually made Posner molecules with lithiums in them. So I'm just modeling it theoretically, and I want to see whether having this lithium in the Posner molecules changes the entanglement, and thus changes the calcium production and the firing of neurons. And I wanted to see whether I could explain the difference between lithium-6 and lithium-7. So I have a system with the spins and the lithium spin, and I work out what the entanglement is. And then I do the same with the lithium-7, which has a different spin, and see what the entanglement is. And it turns out that lithium-6 generates a much bigger magnetic field than lithium-7. So this could explain why you have these different effects from the different lithium isotopes. But yeah, as I say, it's very, very, very. Ideally, I'd like to find a lab that can make me some lithium-doped Posner molecules to test.
Okay, perfect. I think that concludes our question asking or Q&A session. Yeah, we managed to get through all of our questions, so that's great. And thank you again for our attendees also who came on screen. It's very nice to have this sort of interaction with our speaker. So I'm just going to finish this up quickly. So first of all, as always, we really want to thank Betony for this awesome presentation. So please give her a virtual round of applause in the comments and show some appreciation. And then thank you again to all of you for attending. And again, like I said, thank you for our attendees who were willing to come on screen. Then also remember to follow Nova on Twitter and to keep an eye out for our survey. So the survey following tonight's talk will again deal with the bit of connectivity issues and maybe the sound issues that you might have experienced this evening. So please do take a moment to complete the survey and let us know how we can improve for future events. Then lastly, we also want to thank our sponsors, Stellenbosch University and the Biomedical Engineering Research Group. Yeah, and then we will see all of you hopefully next time for our next event on 17 June. Thank you and good night.